Okay, and we're recording now. Okay, and so welcome everybody who's coming in. I think we've got 43 people, 44 attendees now. So people are still streaming in a little bit.
Yeah, good evening, everybody, and thank you for joining us. We're going to wait just a minute or two more to allow everybody to get connected before we start here. Hello, for those of you who are just logging on. If you're looking for Café Scientifique and Helen Mayberg's presentation, you're at the right place. So we're going to give people just one more minute and then we'll get started.
We should have some music playing.
Okay.
Who's got a Spotify account up and running?
It's your cafe.
We should have talked about that ahead of time.
There's some sounds of the bison on the range.
Yeah.
Yeah, I should have brought kind of like sound effects. So, all I've got is sirens. I can hope that we aren't interrupted.
All right, well, I think we will get started here. So welcome to Café Scientifique. I'm Brian Bothner, and I'm the director of Montana INBRE. And we're proud to present this Café Scientifique. And this forum is designed to bring science into the community by providing a relaxed setting for people to learn and talk about current scientific topics. And so the format here is that we're going to have a presentation, and then we're leaving quite a bit of time for conversation, questions, and dialogue. And really, our goal is to bring, leading edge science and the scientists who are doing that work to the public, and allow these fruitful conversations to take place. So this is brought to you by Montana INBRE. So Montana INBRE is a program sponsored by the National Institutes of Health, and INBRE stands for Idea Network of Biomedical Research Excellence. So in a way, this is your federal tax dollars at work. And this cafe format, this is one of my favorite events that INBRE puts on, because it really tends to stimulate a lot of discussion and really, as we said, to, bring science out to the community. Now, this night in particular is a little special because not only is this event sponsored by Montana INBRE, but it's also co-hosted by MSU's Center for Mental Health Research and Recovery, as well as the Office of Research, Economic Development, and Graduate Education. So I'm sure that many of you have been to our live cafes, where we've typically held them in the Baxter Hotel, and we have hors d'oeuvres and people have their beverages of choice, and it's quite a nice atmosphere. We're of course a little bummed that we are not at the Baxter this evening, but we really couldn't go the fall without having a Café Scientifique. So, this is a new format for us, and I'm sure it's a new format for some of you. But I think one of the neat things about this is rather than people having to be in one location, we have people logging in from all over, Montana and, all around the country. So I think that that is kind of neat, and that we're really, essentially extending our Montana INBRE network a little further. So we're going to have the presentation by Helen Mayberg, and this will be approximately 35 to 40 minutes long. And then after that, we will have our question and answer period. And so you'll be able to type your questions into the Q&A button. So if you look at the bottom of your screen, you should be able to see a little Q&A button, and you can type your questions in there, and then we will, I'll be scrolling through and looking at those and we'll get to asking those questions. So I would now like to pass off to the MSU Vice President of Research, Economic Development, and Graduate Education, Dr. Jason Carter.
Thanks so much, Brian, and thanks to you and Bill for adapting and still continuing to host these. I attended 2 last year and really enjoyed them. I keep looking for my drink in the back of the room and it's not there, but we'll adapt with the times. I just want to be very brief and especially make a shout out to Francis Lefcourt and to Michelle Hardy out of MSU's research area. The two of them were really the brains behind this and helped to pull this together. Last year, we started having some conversations about the next steps with the Center for Mental Health Research and Recovery. And Francis shared with us the format that we're doing here tonight of bringing in some individuals in partnership with INBRE. And I thought that was a fantastic idea. And her and Michelle deserve all the credit along with that, along with our committee that is really working hard to redefine our Center for Mental Health Research and Recovery and help it move to the next level. So really happy to be a partner in this with you tonight, Brian and Bill, and just want to thank Francis and Michelle and all the folks that worked behind the scenes to put this together. With that, I'm going to turn it over to Francis, who's going to do our introduction of the speaker tonight.
Okay, thank you, Jason. So it's a tremendous pleasure to introduce Dr. Helen Mayberg tonight. But first, I've been asked to give a shout out, happy birthday, greeting to Crystal Loffer of NAMI in Havre, Montana. So a happy birthday. So I'd like to thank Montana INBRE and all the people who work there and do such a great job, and our VP for Research, Dr. Jason Carver, for his support for this seminar and for the Center of Mental Health Research and Recovery. So in 2014, as a direct response to the high rates of mental illness in Montana, the director of Montana NAMI, Matt Kuntz, and I created the Center for Mental Health Research and Recovery here at MSU. When it came to selecting members for our advisory board, Dr. Mayberg's name rose quickly to the top. Helen graciously accepted our offer and joined us in Big Sky for a kickoff retreat focused on how the center, with its emphasis on basic science and the development of novel therapeutics and neurotechnologies, could best contribute to improving the lives of Montanans struggling with mental illness. As you will see, her cutting-edge work, which has radically transformed patients' lives, serves as an illustration of the advances our center, under the current leadership of VPR Jason Carter and Michelle Hardy, hopes to bring to Montanans suffering from mental illnesses. Dr. Mayberg trained in neurology at Columbia's Neurological Institute in New York, followed by a research fellowship in nuclear medicine at Johns Hopkins. She's been awarded numerous honors for her pivotal work in developing integrative novel approaches to neurotherapeutics, including membership in the National Academy of Medicine, the American Academy of Arts and Sciences, and the National Academy of Inventors, in addition to recognition for her work in neuroethics. She's currently the director of the Nash Family Center for Advanced Circuit Therapeutics at Mount Sinai School of Medicine in New York, where she's also a professor of neurotherapeutics, neurology, and psychiatry. Her goal has been to identify the brain circuitry and mechanisms mediating mood and emotions, with a primary focus on major depression and its recovery. To this end, she's successfully identified imaging biomarkers that define treatment-specific depression subtypes and invented and developed deep brain stimulation for treatment-resistant depression. Based on these major achievements, her mission now is to advance precision surgical treatments for patients with complex neuropsychiatric disorders. So please join me in welcoming Helen virtually, unfortunately, but we're just happy to have you here with us to Montana.
Thank you. It's really a pleasure to be here and be with you. I feel like I'm with you because I can see part of you, and I appreciate the audience tuning in this way. I think this is the way we do this for a while, and I hope you find the presentation as satisfying in this way, hopefully with a good beer on the side, as well as learning something about how we think about depression and how we take care of people with extremely severe depression. I want everybody to appreciate my background. My husband is a proxy Montanian. He lived in Whitefish for several years back in the late 90s, and some of my best memories were in traveling in Glacier Park and in Whitefish and in the whole region. And we'll hope for another trip where we can actually be with you in person. So with that, I'm going to share my screen and put my slides up and get started. Just give me 1 second. One more change. And we're ready to go. So I want to just start with a few disclosures because I am NIH funded, and so this is an important thing for everyone to know. I'm going to be talking about the use of devices in the brain, and these are experimental treatments. And we have these devices donated by manufacturers, Medtronic and Abbott, who really help us to do this research. We need to register all of these studies with the FDA so that everything is supervised to make sure that we're doing what we think we're doing, and that always with safety of patients in mind. We have funding from various foundations over the years, and now more recently from the Brain Initiative at the NIH. And the work has led to patents that hopefully will lead to testing, further testing of what I'm going to tell you about by Abbott for the purposes of, can it actually be scaled to affect more people than the people that just work and participate in our studies. And to that end, I do consult to Abbott in that regard. But most of everything that I'll tell you about has to do with our own funding, with foundation and federal funding, and doesn't involve any other interactions with industry. So let's get started with what we're actually here to talk about. We're here to talk about the problem of treatment-resistant depression. And this is a huge problem, and luckily doesn't affect the majority of people with depression, but depression is so common that if 10% of the population at any time have a major depressive episode, most people get better with the treatment, but about 10% of people fail to respond over time to any of the treatments we have, any of the evidence-based treatments, whether it's a psychotherapy, a medication, a non-invasive intervention like transcranial magnetic stimulation, or even ECT. And if we read and listen to what William Styron, the great writer, had to say about his own depression in his fantastic and very memorable memoir on depression, it's that the state of being treatment resistant is one that no one should ever experience. Because I think, as you'll see highlighted in red and in blue, the idea that no remedy will ever come and that you have a mental pain that's really unrelenting, what must that be like? And I want everyone to really think of the idea to be in that pit and not be able to get out. That's the starting place for what we're talking about tonight. And the way we're going to talk about it is not all of the available treatments, which we can talk about at the end if people are interested, but the idea of how do we leverage a technology that is not novel in the fact that it's been around since the late 80s, but the use of targeted electrical stimulation of the brain with a brain implant. And as the title had suggested, really a brain pacemaker. And the reason to call it that is because a small battery can deliver very exquisitely focused electricity in a very specific part of the brain. And that this device is basically a small wire encased in silicone. It has 4 places on the edge of it. It's really no more than a centimeter in length. This wire, you can see the proportion, is attached to a pacemaker. And through a small opening in the skull, no bigger than the size of a quarter, actually now it can be done really the size of a nickel or even a dime, that with image guidance, a surgeon with a patient awake or asleep can actually place this device exactly with pinpoint accuracy in the brain and actually hook it up to this battery pack to deliver stimulation on demand. So what is that like and what would that be like? I'm just going to give an example first of our motivation to do this back around 2001 was really straight off the back of what had been going on for about 10 years in Parkinson's. And in Parkinson's, where you have a disease of motor control, people have tremor, people are stooped, they have trouble with their gait, they're very slow. There was 20 previous years of figuring out a circuit that regulated that activity. And if you have a circuit, it means you have nodes in the brain that are connected by white matter, kind of the wiring diagram of the brain for movement. And what was cleverly done was to target, through neuroscience research, a particular target at first in the thalamus and then later in other parts of this motor circuit to actually interrupt the abnormal movements that are present in Parkinson's disease. And I'm just going to show you, because this was the motivation that if we had a circuit for depression, we might be able to target a very specific location in that circuit, taking the very same approach that they did in Parkinson's. So here's a woman who has Parkinson's disease. She was seeing actually my surgeon that I work with in Toronto. And here's what happens when she has the stimulator in her subthalamic nucleus and it's turned on and off. And you can see in real time what happens when you apply very exquisitely focused stimulation to the brain.
Able to touch my finger?
Not with the right hand side.
Okay.
I can't.
Can you try to touch my finger? That side's a little better.
That side is better, yeah.
Very good. Just turn it on.
You can see exactly how as soon as it was turned on, the tremor stopped. And actually we can look in the brain and actually see that signal in the subthalamic nucleus that when the stimulator is on, here this area is blue, you turn it off, and here's an activity in a particular frequency in the brain of the oscillation, it's called, in the beta band. It comes immediately back on. So if you think about her in reverse, when the stimulator is off, this abnormal pattern in the brain is present. As soon as the stimulation is applied, you interrupt it and it stops. So if we want to think about a depression circuit, how would we go about thinking about that when you think about depression with its mood, lack of interest, changes in appetite and libido and sleep, feelings of guilt, feelings of suicide? How is that akin to Parkinson's in any way? Well, with the advent of brain imaging, back again in the 80s, but used for the last 40 years to map out many complex behaviors. One can actually take a picture of the brain resting. And our original, very simple-minded thought was, is again, you read William Styron's description, that depression is kind of the brain being off. In some ways, it's a storm of murk, paralysis, psychic energy not being present. When you actually ask most people about thinking about depressed, you think the blues, you think a negative state, but a negative meaning absent state, hard to think, hard to move. And in fact, what we saw, and many people saw, is here's looking on the side view, sagittal view. This is a measure of brain blood flow in a person at rest in the scanner. And one can see red means activity is present, and less activity will be manifest by green and blue. It doesn't take being a neuroscientist to see that something's wrong with the frontal part of the brain. And the best replicated finding in depression is that there's low activity in the frontal lobe. And the magnitude of that low activity correlates with how severely ill you are. And it kind of is intuitive. The problem is that when you think about the symptoms themselves, there's something, again, as William Styron, but many patients describe, it's not just that you feel virtually paralyzed or immobile or you can't think. You have an active discomfort. It isn't just being curled up in a ball, it's actually falling into hell. And that the mental pain, the sense of not being able to do anything, it's actually a positive anguish state. And so in some ways, there's actually a combination of a positive, almost an overactive, shouldn't be there state in the brain, as well as the inability to turn it off. And we looked at that very explicitly and found that even if you induce a negative mood, usually by thinking about loss or death or illness, that even healthy people will transiently change their brain state, and the magnitude of how sad they are, it goes with decreasing in the frontal lobe. This is now a front view of the brain. But suddenly there's actually an area of the brain that's overactive. And that area of the brain is called the subcallosal cingulate, area 25. That's an anatomical designation that I'll use throughout. And in fact, the state of sadness is the combination of overactive 25 and shutting down your frontal lobe. And that the more 25 is up in this state, the more the frontal lobe is off. It's a small circuit. They're tied to each other. When you feel bad, you actually disengage. And then when you need to get moving again, you get yourself up and going, and your mood kind of dissipates. So that gave us an insight into the idea that if the overactive 25 and low frontal lobe is present when sadness is induced, what happens when you take someone who's stuck sad and you change them by treating them successfully with a medication? And what we found, again, years ago, but so fundamental to the rest of the talk, is that here's the pattern of sadness. 25 is up in red, frontal cortex is down in blue. And when you successfully treat someone with a classical medication, this is Prozac, you actually improve the frontal lobe, it returns to normal, and you down regulate, you turn down the activity in 25. Exact same regions. One can go up in 5 minutes, the other one goes down over 6 weeks, same circuit. And so the idea that sort of this painful state zaps your attention and will and movement run by the frontal lobe and the prefrontal cortex is basically enslaved by this overactivity of 25. And so imagine now the fact that no treatment actually can downregulate that area 25. What's something you can do? And one of the thoughts was, well, maybe you can go at area 25 directly. And this is actually the logic for our first experiment back in 2003, where we basically said, can we target this putatively overactive area 25 in depressed people? And because we can't drug or talk or shock it down, maybe we can go in there directly with an electrode, with this technology developed and used routinely in Parkinson's, and block the activity in 25, just like you saw that lady's activity blocked in her subthalamic nucleus and stop her tremor. So we made an approximation with this blob on our PET scans. We use the atlas to figure out where is that in the brain to get coordinates. And we can, under MRI guidance, made that approximation to place an electrode on each side, implant the device, and turn it on. And again, at the time we said, look, depression takes time to get well. We'll turn it on and make sure nothing bad happens, because it was a hypothesis. Could you turn it on and make people better? It was just as likely that we'd turn it on in that area and we'd activated it and we'd make someone worse. So we had people awake in the operating room to make sure nothing bad happened, with the expectation that they would get better over time. And just to show you what happened in this first group of sick people who had failed everything, including electroconvulsive shock, and who had been ill in this episode around 5 years, having tried up to 17 different treatments. It was unbelievable how really treatment resistant these people were. That over the course of 6 months of ongoing stimulation, this is a graph of the severity of the depression. Over 20 is considered severe. You can see these people started around 25. And actually, even in the hospital, the scores dropped as we turned stimulation on. They left the hospital for a rest after we implanted the battery and were off for a week. You see it came a little bit back. But then over the course of 2 months, everybody was getting better. And then after about 2 months, some people didn't hold, and the rest of the people actually had this robust and sustained improvement over time. So this was dramatic. These people had not been well, and we were getting, without side effects, this surprising but consistent with our hypothesis result. And in fact, then we did the PET scans and showed that our goal to take the overactive 25 and low activity in the frontal cortex, that this was what we saw at baseline in these people, that after 6 months of stimulation, area 25 had decreased and the frontal cortex had corrected. So not only did it match what our idea was, the patients were actually doing very well, which has inspired us to then move beyond these 6 subjects. Now, obviously the goal is to get people well and keep them well. But we saw something when we did the experiment with people awake that totally was not anything we had hypothesized or thought. And what we found is that as we were stimulating on each one of those 4 contacts, people had reports or experiences when we would start the stimulation and turn up the current, not in every place we stimulated, but generally in the same place in each person, with statements like what you see here on the screen. One patient said, "The void is gone." I had no idea what a void was. That wasn't in our list of symptoms. What's a vortex? What hole are you in that you just came out of? People said things that reflected the experience happening in real time. They said things like they felt lighter, less heavy, they could breathe, the room was brighter. They even started to say things as we left the stimulation on for a few minutes. They felt more optimistic, more connected. One person said, "Boy, if I wasn't in this device, I'd walk my dog." Another one wanted to clean their house. People just wanted to do stuff. And they wanted to do it when we were stimulating some places and not others. Now, not everybody had the effect, but when we saw it, it was profound, even if it didn't happen again when we were outside the operating room. And so we weren't sure what to make of it because it was very idiosyncratic. But what we missed at the time, because we were so focused on safety and long-term effect, that this was a really, really important clue. And I want to show you what we saw in the very first person who described a void. Here she was the day 18 hours after surgery in the hospital with all of the team just talking with her about the experience. And here's what she had to say. She's talking to the neurosurgeon. I'm sitting to her right. And you'll just hear her describe on the left, this is with the device off. And on the right is what happened when we actually turn it on over the course of about 20 seconds.
And that sensational void, what would you rate that as? Like from 0 to 10, from being the most severe void?
It's like 5. Because in many ways, it's, I don't know what to describe, it's like a nothingness. You know, people talk about the black hole, the black depths of depression, the black hole of it.
You're still in that hole, do you think?
I feel that way, like I don't feel, you know, I don't, yeah.
So you can hear, a little bit hard to describe it. The look on her face doesn't really require words. So if you couldn't hear it real well, you don't need to. Really hard to describe it, but it was this void. So here's what happened when we turned it on, just like we had in the operating room. And you can listen to how she looks and what she thinks.
Well, I feel calm and harmony. You know, I'm feeling like I want to smile or laugh or joke around. I'm looking at everybody's expressions on their faces. And
you feel more in tune with the group?
Yeah, more in tune with the group, looking at everybody's expressions. And
So funny, the reason I asked her about looking at everyone's expressions is there's 3 totally exhausted neurosurgery residents sitting in the room, having been dragged by Dr. Lozano to watch this, totally uninterested. And they're sitting there, and as we turn it on and she kind of emerges from that state, their mouths are hanging open and she's watching and is amused by their reaction to her. And when you think about the nature of the disconnection that depressed people feel for other people, you look at this little video and you're watching depression change the first state in real time. And so I always go back to this case because it isn't that we ignored it. We didn't realize how important it was, and I'll get back to it in a minute. So I moved from Toronto to Atlanta in the middle of this experiment, and I was going back and forth. And when I got to Atlanta in mid-2004, the only person I had to work with was a neurosurgeon, Bob Gross. And you can wonder, like, well, why did I move from Toronto to Atlanta and abandon my other small team? Another story. But over time, we recruited Paul Holzheimer from University of Washington to come work with us. And he was interested after he read the first paper. And over time, over 10 years, we built a team. And I want to emphasize that this kind of work, once you get your foot in the door with something new, it isn't about a psychiatrist, neurologist, neurosurgeon. You need people to care for patients, to monitor them. And most importantly, you can see the clinical team is small. The scientific team grew because as people saw what was going on, we were all trying to figure out what had we done and could we understand the mechanism to not only understand what we were doing, how to make it better, but what did it teach us about depression itself. So again, one of my only graphs is to help you know is how do we know that people are getting better? Well, in Toronto, we went from those first 6 to a total of 20 in the first group. And as you can see from, again, tracking the depression rating, the dotted line is when you cross that line, you're officially a responder. when you get down to a score of 7, you actually are considered well in remission. That what we found is the additional patients followed that same trajectory. By about 2-3 months, they were seriously well. And as we followed that group in Toronto over years, this is Dr. Kennedy, the psychiatrist that followed them. If you got better, you stayed better. And this was now out to 6 years. And think about it, that most people, if they respond to ECT, they're guaranteed to relapse within 6 months if you've reached a point of having failed 4 other treatments. So the idea that with this device, this pacemaker, we were holding the brain in a well state was really something that we hadn't seen before. And in Atlanta, we expanded what we did with Dr. Holtzheimer, and we also studied people who had a type of depression, bipolar type 2 depression, because they're notorious for when they respond to a drug that they can flip into hypomania. And the area that we're stimulating in the brain is an area that's enriched in receptors that bind serotonin. So we wanted to know if we were stimulating in that area, would it be problematic for people who have bipolar type 2? And the good news was that people who were ill a minimum of 2 years and stuck depressed, if they got better, we could not induce any kind of dysregulated mood. So we couldn't make them hypomanic like medicine. So it's working differently than medicine. But the good news and the bad news, or the bad news, and to me it was good news, unlike Toronto where most people got better, it was very hard to learn about what non-response meant. In Atlanta, we didn't do quite as well. At 6 months, our patients, we only had 40% of them better. But as Dr. Holtzheimer, with trial and error, changed these settings, he was able to get patients mostly well, 90% response rate by 2 years. So we had both replication, extension with the bipolar 2, but a new important thing to drill down on, which is what explains non-response or what explains a delayed response. So when you get into discussions about non-response, because most treatments, some people do great, some people do get some effect, and some people do terrible. It's always like a 30-30-30 percentage. You really have 2 main categories of what accounts for non-response. And it really comes down to who do you want to blame. Blame the psychiatrist, got you the wrong patients. And we all know that not everyone responds to every treatment. And that's not a question we actually can answer. We don't know how to decide what medicine. We don't know how to decide who gets therapy and who gets drug, another topic of discussion. But the one thing we can control is trying to figure out if we're in the right place. That's something we can adjust, if not in the same person, in subsequent people. And so that became the focus of our attention. And again, you have this electrode, you're trying to aim it at this tiny spot, but in fact, it's really not the spot we're aiming at. What we're aiming at is spaghetti junction of wires in the brain, the white matter connecting area 25 to a whole group of other regions. So back to that kind of very simple depression circuit, it's not just about area 25 and the frontal cortex, it's area 25 and some other stuff. And the question is what other stuff? And one can make some real estimates from the literature, but mostly done not in humans, but in non-human primates, where people have mapped out the anatomy. But the question is, this is a picture, this colored picture isn't a drawing. It's actually a rendering of a type of MRI scan called a diffusion scan, and lets us look at the track map, the white matter tracks. And that's something that then we wanted to do. So what was a lucky break for us? Well, it wasn't a lucky break totally, because what focused our attention on the tracks specifically came from an observation in a young woman. She was only 30. She had been disabled for 8 years. I'd actually met her in Canada. that she was afraid to be in the first study of 6, that because we hadn't done it before. And she read about the study and 8 years later contacted us in Atlanta to see if she could participate there. So an 8-year delay, even from the time we had met her. And she was what we thought was an ideal candidate. We did the implant. And as you can see from her graph, her score is going down. But when we got to the end of 6 months, she really didn't, she had a very partial response. And if you asked her how she was doing, she still had this real painful noise. It was really almost like it was like a physical noise that made it hard to move through. And she tried very hard. She really couldn't do anything. And when she got to the end of 6 months and wasn't well, you can see her score actually got worse. She was trying so hard. And we went back and looked at the scan and said, you know, from all the patients we'd done, and she was patient 17, and we had some that were really robust responders, we were starting to see that you could even see on these MRIs, these white matter fingers. We hadn't even analyzed the tractography yet. And it seemed as though we didn't have choice to stimulate her a little lower in the brain. So we all discussed it and said, we think enough that we know we're in slightly the wrong place. Let's re-implant that side. And the surgeon thought we were nuts. He says, it can't make that much difference, a millimeter and a half to move it. But we moved it. We were in the operating room. She was awake. And when we stimulated this third contact, which is literally just a little lower than the lowest one where it originally was, she suddenly had this awakening with the noise just turned off. She felt lighter. She felt connected to the people in the room. Things came into focus. She started talking faster. And we really knew for the first time now that what seemed like so similar was actually so different. And what we then embarked on was to say, what did we do different in the track map? And in white is what we were doing to start. We were getting the cingulate into the thalamus and a little bit into the frontal cortex. But when we moved it, we got much more into the frontal cortex. And when we looked at her, as well as all the other people who had done well, we actually found that it wasn't a spot, it was a nexus, a hub of 4 very specific white matter bundles in the brain. And you can see a map. It's now a template that we use. You have to hit the cingulum bundle. You have to hit this bundle called the forceps minor that goes out to the frontal cortex. You have to hit this bundle that goes to the amygdala hippocampus. And you need to get those bundles, these small ones that go subcortically down to the basal ganglia and the thalamus. And suddenly we had an explanation for non-response that didn't require us to pick different kinds of patients. And we could actually adjust based on these maps and convert non-responders to responders in most cases. So we have this map. I'd drawn it out. You know, we knew the connections, but what we needed was to be able to actually convert that to a scan. And now what we can do in every single person, we can drive around on their individual tractography map beforehand, find the exact spot that hits those 4 bundles and implant with precision that the surgeon possesses and actually have the capacity to stimulate where we want. And what we found by this evolution of kind of guessing the approximate area that we started with to actually using this template to decide exactly where we are and never change it was that instead of having a slow decrease in the score over time in red, the green is what happened by just using the individualized map to guide the surgeon. Not only did we get better faster, we got better more robustly, and we had an 80% response rate rather than a 40% response rate with less work. And now what we're doing with giving more stimulation in the operating room, we're getting an even bigger and more robust effect by boosting that initial signal. So I want to show you, though, that, you know, it isn't just that you get better fast, but that there's an evolution of the recovery even when you control for this. And this is one of our patients, Brandy. And I use her name because she has been an advocate for herself, and she is open about her implant. And I want you to hear her description and her trajectory to kind of see what we learn and what we continue to learn from the patients to try to figure out how to map what we're doing. Because what's happening now at INBRE is we now have, you know, over a 200 patient year experience that basically if people get better, they stay better. So understanding the trajectory, helping it to go faster, knowing what's happening at different points in time is really important. So here's Brandy before surgery, then in the operating room, and then describing to the psychiatrist at 2 months in, and then 2 years in, what her own evolution is of her recovery. And I'm just going to play them in a row.
For me, it's like a stickiness. It kind of holds me down, and it takes so much effort to do anything or to experience anything. And there's always that cost of, it kind of reminds me of like scar tissue that, you know, every time you stretch it comes back and it holds you even tighter.
It's a really amazing thing to listen to these patients make metaphors of this state. I mean, she doesn't look like she's in pain, but she sort of does. What is it to kind of be entrapped by an alien that you feel like you're being held in, but you're not? And it's quite profound, and it's very physical. And when she's in the operating room, you can see the situation in the operating room, how difficult it is to be in the operating room and then expect a patient to be observant enough to be able to tell you what's happening. But you can see what happened to her, and it's written underneath so that if you have trouble hearing it, it's transcribed what she said.
Patient ratings, one sound, stay on.
Got stronger. Describe what got stronger.
The sensation of being lighter.
Try to give me a
I
little
know,
more.
I am like physically not so much surrounded. It's, almost like more air in my body.
Mentally, is it have any mental qualities to it, or is it still mostly physical?
Actually, last night it kind of was, there was that lighterness of my mood that went with the lighterness of my feelings.
I'll tell you, as a scientist, everybody will be scratching their head. What does that mean? But as you listen to it, one doesn't doubt that someone's experiencing something and trying to describe it. But as you get to 2 months, she is more reflective of what that was.
What thing did
What happened was
Hold on, sorry.
incremental changes. Things got,
Sorry.
what thing did
what happened was
stimulator do from before to after?
incremental. It gave me the break. It gave me the energy. It gave me the release from the oppression. You know, it made things a little bit easier. That's what it did, is it lifted enough of what was holding me. It just released it enough for me to be able to wiggle and release them a little bit more, and it just kept lightening the bombs. That's what it did, was it made it possible to breathe again. made it possible to move again. It made it possible to open my eyes.
Again, very physical. And I always, every time I watch it, I'm just moved by her description. So here she is 2 years later. She's back as a consultant, had a relationship, you know, busy, goes to Comic-Con and wears a cyborg t-shirt. And here's how she describes the evolution.
Things got a little bit easier. And even in the smallest things, it got a little bit easier to brush your teeth. It got a little bit easier to get out of bed. It got a little bit easier to have hope. It just started a cascade of positive instead of the cascade of negative. And that was the difference. DBS didn't make me happy. DBS, it didn't solve all of my problems with how I was thinking or how I was behaving or my lack of boundaries and relationships. It didn't solve any of those things, you know, that needed to be worked on. All it did was make it possible for me to not have this huge burden preventing me from being able to solve all of them.
So you can see there's an evolution, a fast effect, a slow effect.
What thing?
Hold on. And that this becomes the focus of all of our attention. How do we understand what that rapid reset is that now is predictable by the track map? We can say we've done 21 out of 21 patients and we know precisely which contact will affect that first change. But then there's this slower effect that in some ways is unpredictable in different patients. And they say the darndest things. I mean, here's a quote, and we don't have the quote from Brandy complaining about how hard it was for a period between 2 months and a year, where patients didn't realize until they were out of it how much work they need to do themselves. So a lot of our work now is to try to figure out what are those stages. And for the basic scientists, we look at this in models like ketamine, where you can get a very rapid effect, and animal models can show that you have hiding spines in your brain on the ends of dendrites, and they pop back out immediately, you know, a day later. What do spines do? Something very fast. There are other things where you can show in animals that if you stimulate, you activate the very cells that make white matter, and that you turn on those oligodendroglial cells and they make myelin, and myelin can then make connections stronger. And we talked about the fact we're stimulating white matter, and that may be, and that takes time to make the white matter. The third layer is we may be getting changes where you have to relearn, and actually you may actually grow new neurons that actually allow you to set down new habits. So that these fast effects and slow effects, there are other neuroscience models that might make us understand it. We can't study patients quite this way, but we can develop collaborations with animal researchers to do that work. But what we can do is we can put people back in scanners. You can't put them back in an MRI scanner to see if they grow more white matter because you get kind of noise from the electrodes themselves. But we can put people back in the PET scanner. And we knew that while we had these remote changes in the brain, people who didn't get better didn't fill in those areas. But we really didn't know what was happening fast because we didn't do scans until all the action was over, because we didn't know that we would need to be looking really early. So what we started to do with Dr. Choi, who had done all of the work with the track maps and worked out that template, is we use techniques with PET scans, and we break the brain into modules, into circuits. And we ask, how do different systems in the brain change as we're giving stimulation? What happens as the reset? What happens with chronic stim? What happens with early stim? What happens with late stim? It turns out that not all the systems in the brain change. Some systems go up, some systems go down. But we found that one system that, interestingly, but maybe not surprising, the default mode network of the brain, kind of what you do when you're resting, when you're not doing anything, it actually overlaps exactly what we stimulate, particularly the front part of the default mode. And it turns out that if you look at this system in the brain is tracking that depression recovery signal. And that if you look at what happens early with changes in this system and what happens late, it turns out that early, you turn the system down. You care less about yourself. And then late, you turn the system up. And it's actually the back part of the default mode where you actually may be projecting yourself outside yourself as opposed to internally. That's an interpretation. But just by following the phenomenology of the patient, we can actually drill down on the neuroscience the best we can. So the last thing is, what is that reset about? You can't do the PET scan in the operating room. And if you move out of the operating room, it seems to already be gone. It's not like Parkinson's where you turn it on, you turn it off, you turn it on, turn it off, and you can do it as many times as you want. You don't fix it. Something about depression fundamentally changes the first time you turn it on. You're not better, but you're different. So how do we use the patient's own metaphors to track what's happening in the operating room to know what we're doing where? And again, Dr. Choi with the imaging, Dr. Riva Pose with the behavior, we took those funny statements and we started to try to catalog them. What's the nature of the statements? And when they say certain kinds of behaviors, things that are really about the body, you're always hitting one of those 4 networks, the cingulum bundle. If you hit the forceps minor, the very area in that one patient I showed you that we had the incomplete effect, that's when you get these complex, you feel connected, you feel like doing things. So we're even able to break down in our circuit what part might be contributing to the very behavior, but you don't just want to be able to breathe, you want to be able to run because you can breathe. You know, it's being well is the integration of your function to kind of do whatever you want. So we now, as a first experiment, and this is really very new, we now are measuring the electrical activity in the brain when we turn it on those good contacts for the first time in the operating room, not just listening. And when we turn it off and we look what happens to the brain before versus after good stim, we actually now have a signal of how the brain signature is changing. And it actually isn't all that much different from what the Parkinson's patient showed, except it doesn't go away. We see this decrease in the signal from before we start any stimulation to when we're done. And this red line is dropping in the frequency of what we call the beta band. It's beta and gamma. All these patients, if we look how they're doing a week later after exposure in the operating room, no more stimulation, they all have dropped their depression score, all of them. Some of them more dramatic than others. And the magnitude of how much this beta decrease is, the better they're doing a week later. So we're actually getting a notion of what the first effects of this depression recovery is. It basically over minutes. So just to kind of finish, you know, if we're dependent on the self-report, what we notice is patients go through these stages. They look different, and then they do more, they move more, and then they don't actually feel better till late. So if your rating is always dependent on what the patient thinks going on, they don't actually know how they're feeling as their brain is going through this recovery. And you can see it on this lady's face. I mean, you don't need a scale to know that here she doesn't look so good, and here she looks pretty awesome. And we need a way to actually just quantify what's totally obvious. And we need to know, because when you look at these scores, here's someone that, wow, they did great here. And all of a sudden now the score is getting unstable. You know, they having some life stressor, you know, trouble with their kid, lose their job, but this looks really bad. Maybe we need to know here if it's a signal to turn it up, or here to turn it up? What should we leave alone? What should we have you start therapy? And what is something where we need to make adjustments? And the score is actually confusing. And so what we're doing now is experiments now with my new team at Mount Sinai, is we have a prototype device that instead of only being able to do those recordings on the brain at home, the device itself has a chip, and it means that we can do telemetry. We can track the brain signal in area 25 all the time. The patient actually, here she is in bed after surgery. She's got a tablet. She can record from her brain. We can record her face and get an idea of her facial expression. We can do ratings of her mood so that we can do analyses of all of these things in an integrated way. And we can even in the operating room, instead of just having these utterances, we can actually track the facial expression just like you saw in that first patient 0, except now we can quantify it. And here you can see a set of muscles in the face right before we gave the good stim. And even if you can't see the picture, we can see a change in the activity of those motor units in real time. So we have what the brain does, we have what features of the face does, and now at home, the patient can be recording herself and we can get ideas of how she's doing, and we can look for these same motor units in videos at home. We can look at voice, all the things that hopefully everybody has heard through this of clues of what parts of the brain convey our emotional state, our voice, our heart rate, our expressions, not just how we say we feel. So to end, the last thing I want to bring up, because there's the neuroscience, there's the clinical science, and then there's the ethics and the experience for the patient. It may be a cool experiment, but we got to make sure that everybody is aligned of what the device does and what the patient has to do. And when people have been sick a long time, they're not sure what's for them and what's for us. Neither do their families. But what we want is obviously a sustained, durable recovery. What could be worse than to come out of that state and be doomed to go back into it? Patients are terrified for months that feeling bad in any way means they're relapsing. And to get over that requires rehab, to trust what's happening, but importantly, to learn that having a bad day doesn't mean you're relapsing. Having a bad day is just having a bad day. And everybody needs to learn that, and we needed to learn that. But I think the more kind of meta comments and existential things are, for a patient, who are you without your depression when depression has been what defines you? And as one patient sent me in this lovely little card, was that, life's hard. And I think we're all experiencing it now, how hard the world is, even when you're well. And the patients learn over time that they're lost, but they're trying to figure it out, and it takes time. And gee, I'm somewhat happy. I'm definitely overwhelmed, but importantly, I'm not sick. And patients come to learn to tell the difference. I want to end with this last thing to think about, because people are always a little wigged out no matter how well you watch someone recover and how well they look. Many people become unsettled by the idea of somebody having a machine in their brain. What does that mean? Are you making somebody better than themselves? People are always caught up in augmentation. Where does this technology go? So Brandy took on an assignment from me. I got called by the BBC to basically be punked on a show to talk about digital humans and about, and it was an entrapment. And I put it on her because she likes talking about having the machine. And she was on a segment, with some scientist that likes sticking gadgets in himself so he can track himself. And it's kind of strange. but she was on this show where basically the host was really pushing at her to talk about being a cyborg and was really trying to get a rise out of her. And I just want to end with watching what someone takes on with this. And I think it's, in some ways, it's the most important part of realizing how we utilize technology as it's now evolving at a kind of mind-numbing rate. So this is, you're going to hear the interviewer, and the question is on the nature of being human. And then I'll close with her comments on being a cyborg.
Cyborg, when you forget the technology is even there, once that transformation is complete, are you more or are you less human? For Brandy, the answer is clear.
Being a cyborg absolutely restored my humanity. Depression had wiped out my personality, my ability to cope or function, my ability to interact with people. And becoming a cyborg has restored my ability to be a member of society and to have compassion and empathy and to interact with people. So I am now part of the people around me because I have this implant.
And lastly,
I am a cyborg for the rest of my life. And I'm okay with that because it gave me a life. But it is something that is part of my identity now.
so I'm going to stop there. And I have this slide that if anyone in the audience feels connected to what's gone on, the resources in Bozeman and the Bozeman area in Montana are vast. And I think anyone on the team that's hosting this is certainly where you can reach out, but here's some basic numbers, and I'll stop there.
Wow, thank you very much, Helen. That was, I want to use the word, it was a little mind-blowing.
Good, I was successful. Okay, I'm glad for somebody. Okay.
Yeah, I've got just, wow, so many questions. But so is there someone on the video feed that has a question that they'd like to start with?
I'll ask if anyone else.
Yeah, go ahead, Jamie.
All right, okay. So I had a question. So without getting into the weeds about like stimulation protocols and stuff like that, so I was wondering whether you think, so presumably if people are doing well, you can't really turn off the stimulator under normal circumstances for ethical reasons. So, I mean, is your intuition that there's a threshold, like they improve to a certain extent and then they can carry on their own? Or is it more likely that they will continue to require the stimulation forever?
Thank you for the question. So we have tested it because in fact you have two ways in which we've tested it. One is we had a blinded discontinuation at 6 months where patients knew if they were well, sometime over 2 weeks, they'd be turned off. And actually with a week off, there's no change. So unlike Parkinson's, even early on, if you turn it off, patients can't tell. It makes actually programming hard, which made us very lazy. And we were lucky that we picked a high frequency, we didn't mess around with it, and we turned up the current a little bit. The other more important kind of naturalistic discontinuation is in the old systems, it was a primary cell battery, which means that at some point it would die. And we learned that it could be anywhere from 4 years with the original cohort battery to 2 years in the early INBRE cases. And so you learned what it was like to get the dwindles. So even when the battery died, it wasn't that rapid shift. It was a slow slide. People just started to wake up and kind of have trouble getting out of bed. And then once they started to worry about it, it could catalyze. So people can relapse. And if they have a broken device, they won't be right, and it has to be fixed, and the devices can break and have to be replaced, or the battery dies, or it's a machine. There are things that happen, but different places in the brain that are being stimulated. And I wanted it to be experiential. It was already too long, this talk, but we are not alone. People are stimulating in the medial forebrain bundle. They're stimulating in and around the nucleus accumbens. For the neuroscientists, it's areas that are known to be in the reward circuit, and people get happy. And when you turn it off, they have a mood crash very fast. So again, do I think over time that you could turn this off? We have people who have devices or batteries go wrong late, and it doesn't seem like they can, but I wonder whether or not we could condition people to tolerate the discontinuation. And I think we need a readout to know what we heal and what we don't, and we don't know. So the good news is people don't develop side effects over time. The same setting is what they do indefinitely. It's almost like it just is incorporated into their rhythm, and it keeps them from decompensating. I think that's why I don't call it a pacemaker except for the title, but in some ways it's behaving like a demand pacemaker, but you don't know what the arrhythmia is.
Thanks.
I'd like to ask a question. As you undoubtedly know, that suicide is a leading cause of death amongst American Indian people. Does your approach potentially have some application to dealing with the factors that lead up to that problem?
That's a complicated question, so let me break it into a couple of parts. A, disclosure, because we didn't know if the technology would work and what the rate of change would be, we never enrolled people who were actively suicidal. And that, the best treatment for that is ECT. And parenthetically, what may be a very good treatment for active suicidality is ketamine, to disrupt that acute can't stand it anymore. All of our patients in that hopeless state where you're that resistant kind of feel, they will all endorse that it would be okay if they were dead, because what else is there? If I'm like this forever, that wouldn't be bad, but they are not actively suicidal. And I think that nuance is really important. That said, suicidality is multifactorial. And while a big cause of suicide is major depression, it can be orthogonal to being depressed. And even, the idea that, when you think about Brandy or you think about these patients, you have this uncomfortable feeling and you can't move. That's kind of protective. If you have no motivation to get up, you can't hurt yourself, even if hurting yourself might make you feel, you might think you might feel better. And so the danger, the care to have someone who may be actively suicidal, who may suddenly be able to move, but the mood is not quite clear, I think I would be afraid of that. But I think that for the population that you're referring to, the other contributors to high suicide rate, whether or not that's responsive to pharmacotherapy or this, I think is an open question. But the problem definitely needs to be addressed.
Jason, did you have a question?
Yeah, real quick, because I can see you got a lot of Q&A questions coming in, Brian, so we'll get to those. Real briefly, any subjective or objective measures of sleep that your lab looks at, and did you see improvements in either short-term or long-term sleep with these individuals?
So great question. So we attempted to do formal sleep studies, put people in the hospital, turned it off, and were kind of very frustrated because they're on medicine that we add the DBS to, and the medicine variations really made the small sample analysis really hard. Anecdotally, there are patients who feel quickly like they sleep better, and the projections of area 25 down to the hypothalamus would certainly suggest that sleep might be affected. But on the other hand, we have patients where their only point on the Hamilton over time is a sleep problem. So separating the depression-related sleep, which seems to get better, from a primary sleep problem, we don't do. Same with anxiety. If someone has an underlying anxiety problem that isn't just the best descriptor of that interoceptive state, you have to treat the anxiety after the fact. That's not effective. This seems to affect that psychic pain movement system, and everything else seems to follow along.
Okay. Matt, did you have a question? And then I'll move to the ones that have been written in.
Helen, thank you so much for coming back. I'm sorry, my internet was a little bit spotty during the question and answers. That's from living in a cabin in the woods. I have a question about, do you see any hope for biomarkers for bipolar disorder after all of the imaging that you've done? We have so many people that do react negatively to antidepressants, and I'm just wondering if you see any hope for that.
Well, I'm an optimist, so I always have hope. The realist side of me says, as we discussed when I visited, 6 years ago, how do we get the field to help to address the pragmatic questions and not only address the esoteric questions? So imaging, since I was visiting, we continue to look for predictors, not of the bipolar, as I know you're interested in, but in just to select and get people to the treatment that's best for them. So if you need therapy, get a therapist and never see a drug. If you need a drug, don't be afraid and get the drugs so that you get the treatment that's best for you. I think that the bipolar, unipolar approach has been more about diagnosis and not within the bipolar to understand who might be at risk and how to best choose treatment. So I think, again, the lobbying required, or the scientific lobbying, is, imaging is always going to be the sexy start that has to be converted to something more accessible and scalable. And so it's going to take people really focusing on your question, getting a biological marker with imaging that can then translate to something that everyone can do in the field. In the same way that our work may teach us about what we do and where we might focus in certain kinds of patients, that obviously everyone can't have an implant. If we can figure out what we're fundamentally doing, how can technology 10 years from now be doing it without going into the brain, that we'll have ways to get at it that you could do at home, set up your own device. So I know was taking the opportunity to remain optimistic while being sort of disappointed that we haven't answered that question.
That's perfect. Thank you. And thanks again for coming.
Nice to see you, Matt.
Okay, Helen, I'm going to now go through the questions, and you have generated a lot of really interesting questions, and I think I'm going to combine a couple of questions here. And I think one thing that sticks out to, I'm sure many of us who are not neurosurgeons, is the risks and the precautions associated with this. I mean, no matter how small these electrodes are, it seems like they're, what are the risks of doing this? And then also, you talked a lot about the positive outcomes. I mean, are there cases where there's a dramatic negative outcome?
So let's talk about risks of surgery. And again, I'm not a surgeon. I actually am personally, believe it or not, quite risk averse, so that I don't take, as a neurologist, you have a break in the skull and you get anywhere near the brain, it's never a good thing. Okay, so that staying out of the brain is always the choice. And that's why we focused on people who had really exhausted all possibilities. Now, this kind of brain surgery relative to other kinds of brain surgery is quite safe. But like any brain surgery, you have anesthesia, and you can always have trouble with anesthesia. You are putting something in the brain, even if small, and that there are vessels, and you can bump into a vessel and you could have bleeding. That's the biggest risk. But the pictures and the navigation software allows a competent functional neurosurgeon to plan the trajectory to get into the template as we described, because it's hitting a three-dimensional object at a point. You may say, well, I've got to go through something bad in order to get there. You can always find a trajectory with work, because you can never go through a vessel. You don't want to go through the ventricle. There are things in the brain, but remarkably, it isn't like you put this wire down and you bump into something and you damage it. And why that is, it just is. The brain, the white matter, there's so much white matter, things move out of the way, and you can get it where you want. And it's a pretty big area. I mean, there are other areas of the brain that putting something in it would damage the very place you're putting it. That's not true in this this constellation of white matter. But people can have a seizure. You know, you're going into the brain, they've had anesthesia, it can be irritable. There can be a little bleeding on the surface. It can give them a seizure. It can be treated. That you could have a stroke if you had deep bleeding. We haven't had that. We haven't had any catastrophes of someone having a cardiac arrest because they're under general anesthesia. Those are all known risks. The biggest risk is that when you've gone into the skin and you've opened up the chest to put in this battery, is you can get an infection. And that can be treated with antibiotics. And I mean, mundane stuff, you have to keep it clean. Or the patient has to be fastidious also. And that's where we've actually had complications. Sometimes you have to actually take IV antibiotics for 6 weeks. Sometimes you have to take the whole system out. We've never had a brain infection. Tends to be local, but the infectious disease people will make you take it out and then replace the battery. So there's a delay. So those kinds of things are there relative to the risk that you might hurt yourself over time. I think there's every benefit has a risk, but in terms of the surgical complications, it's less than you might think. On the topic of, hold on, I forgot the other topic.
Well, are there,
people who don't do well, people who don't do well. So in our experience, we've seen people get no effect. And once we've checked that it isn't where we're stimulating, we've taken it out. So that those are patients where you actually wonder whether or not this wasn't the right treatment for them. That not that they didn't have depression, but that this actually wasn't appropriate. And that's why being able to explain a lot of the non-response by making an adjustment of where, that leaves a smaller number of people where the problem is it just wasn't right for them. So what I feel now is, as I look for these patients that are slow with a lot of negative pain, if people only have inability to feel pleasure, this may not be the best target. What we have never seen is that the stimulation actually makes you worse. And that in some ways, I would have hoped we would have seen that, because it's reversible and we would have learned about something about the stimulation. It was that's in some way a scientific disappointment, but a curiosity because it seems to turn something off, and the rest is plasticity. You know, in Parkinson's, if you get in the wrong place, you can get funny movements, or your eyes go crossed, or you can get anxiety that when you turn it off, it goes away. You bump into the wrong thing. I would have thought that we'd see more problems just with side effects of the stimulation, and we don't. But I'll tell you, to go through this ordeal and to not get better in the way you want is beyond a disappointment. On the other hand, people that ask for explantation often have gotten benefit, but their other diagnoses may be unmasked. So someone with bad anxiety needs to have attention to that, or someone has a problem and having the device they need to pay attention to their other problem. So they just as soon not have the device. And I kind of always advise, even in people that have been in studies, if you aren't getting an effect and it's been maximized with therapy and locating the right place, assume that this wasn't the right treatment for you and get explanted so that you would be available to try something new that might be on the horizon. No one should feel that if they don't qualify, that that's the end of the line, that there are always new things. There are many new things being developed, and hope is important. But I want to make that for people who might, be ill, that hanging in there is really essential. But in terms of what we've seen, it's either does something but not enough, does something and you need to work a little harder yourself with therapy, or you just weren't right, and it's a minority. But I think my team has gotten very good instincts about picking the right patients, and I think we need to understand that better so that that's translatable to others.
Yeah. Okay, very nice. Well, so this is something then that you a question that you kind of got at, is this treatment right? But the question is essentially, how heterogeneous or homogeneous is this, involvement of Area 25?
So for a while, I had hoped that since most depressed people don't have kind of overtly overactive 25, but you have to suppress it with other treatments, I figured the fact that we had this blatant overactive 25 that was going to be a biomarker, that should be the people we enrolled. Right. And it turned out that people do well and they don't necessarily have overactive 25. So that's not an exclusion criteria. On the other hand, we're studying with fMRI the relationship of Area 25 to other regions. And we're almost looking for the disconnection of when Area 25 is active, what is it talking to? And that failure to communicate with other key regions may be a better biomarker. And we're trying to compare our patients who do well with DBS to patients who recovered from ECT, so that you could decide if ECT is for you, then you should get it. We don't know yet is we take people who fail ECT, maybe better to get them before they have to go to ECT, because then they don't risk any side effects from ECT or relapse. But we don't have that yet. I think it would be misguided right now to say it's an inclusion criteria to have overactive 25. But I think the critical thing is you have those connections, and that we can target it in an individual manner.
Okay. Well, now a question, I guess, a little bit more on the technical side is, you've talked about kind of the stimulus and the frequencies and, turning it up. I mean, so how variable is that, and how much does it have to be tuned for a person or a patient?
So very good question. So like I said, we got lucky. I mean, we're just stupid. I mean, sort of like Mr. Magoo. I mean, the Parkinson's people wanted at the time in the year 2000, the mechanism of DBS for Parkinson's what's called depolarization block. You're blocking activity. So it's a misnomer. It's stimulation, but it's actually blocking the neurons. They're not really being blocked. It's just you're stimulating them so fast they can't keep up. So the net effect is a block. Well, that's when you're stimulating neurons. We're stimulating neurons plus the white matter. And it turns out the white matter actually is activated. The question is are you stimulating fibers that are excitatory or inhibitory to wherever they're projecting? But that 130 hertz was the frequency to block, and that's what we thought we wanted. And because it worked, and when we changed the frequency to see if we could make it go the other way, it didn't change fast. We just said, we're going to get all mixed up. We're just going to keep it at 130 hertz. And we weren't doing enough people to say, oh, well, let's do this person at 120 and this person 140 and do what you might do if you were testing a new drug or you were exploring the parameter space, because you have an infinite parameter space. Yeah. You have frequency and pulse width and current. And we don't know, and we don't have an acute signal to be able to tune it like tuning a radio. But I think that now with the new device, as we characterize the acute effect, we can look at how different parameters affect it, because it may be, maybe people can get faster if you just tweak their frequency a little bit. What we do know is if you increase current or change the pulse width, what you do affect is how much of the white matter you stimulate. So the volume of tissue activated can be mapped. So you can know if you're incomplete or not, like that one case. But the maximizing the particular parameters hasn't been something we do. In other things like Parkinson's, they try different things. They give different kinds of sequences. But they also have a different problem in that the brain is continuing to die and that you're always playing catch up to try to be working on something that's going in the wrong direction. We don't have any evidence that the brain is degenerating. So it's almost behaving like we hold it and let it repair, but it's always at risk to decompensate without it. But I think it's a really important theoretical and engineering and kind of electrophysiology question that we don't ever read out. So it's kind of beyond, it just becomes frustrating to try to look.
Yeah. Okay. Well, your mention of Parkinson's disease and also of other researchers stimulating different areas for depression, but there's been a number of questions about, what about other problems? What would be potential likely other places to apply this?
So thank you for that opportunity to do. So already available is stimulating a part of the brain, the anterior limb of the internal capsule, for obsessive compulsive disorder. So again, in theory, any neuropsychiatric disorder that can be defined as a circuit, meaning that there's a set of regions that talk to each other and that there's one region that you think is accessible and could affect change by other information. OCD is one of those disorders and is successfully treated in people who become who don't respond to medication or psychotherapy. And it can be really life-changing for people, can't leave the house because they're washing, or they can't speak because it has to be perfect. And that's available now. But the same issues apply. They know approximately where to go, but they don't know what the exact template is. So there's a lot of trial and error, but that's worked quite well. People use it. There's a lot of work on Tourette's syndrome, which again has an obsessive component. So it has an OCD component, but it also has motor tics. So it's a combination movement disorder, neuropsychiatric disorder. And there is a lot of literature on Tourette's, but it's done off-label. It's not approved and it's all experimental, but there's enough known that people get it done and work to have their insurance pay. So that's one. People are doing experiments to understand intractable post-traumatic stress disorder. And that's work now. The only study that I know of is at UCLA, and it's been going fairly slowly, but they're targeting the amygdala to kind of turn off the fear response system. There are people trying to, one that people have tried to do a lot, but it's been harder is addiction and how to stop craving. So the compulsive, all of the compulsive behavior. So when you think about OCD, you have an obsession and a compulsion. Addiction, you have a craving and a compulsion. Overeating, there's work at Stanford with people who have, have failed, have compulsive overeating and are morbidly obese and can't stop. So there's a lot of logic of these addictive kind of behaviors. They've tended to go into the same area, the kind of reward reinforcement area, and it's been tough. But that work is going on and theoretically should be successful. There's work on pain, because implanted stimulators in the spinal cord can be very effective, but they kind of stop working. So some people are trying to understand how to target the physical pain network. I mean, it wasn't an accident that I was talking about psychic pain and how does affective pain relate to physical pain and what circuits are shared. And so people are looking at that. There are studies now on schizophrenia, but again, the logic of what is the target, people have ideas. So there's a study at Johns Hopkins, small study, and also in Spain. And I think that's kind of the ones I know. I think in the field, it's so, psychiatrists as a group, they're not like neurologists. They talk to people. They don't go into their brains. I even notice in my own team, Dr. Holzheimer studied neurosurgery and then became a psychiatrist. I mean, he had thought about being a neurosurgeon. My other fantastic psychiatrists, there are some who never, always find a reason to be busy and never come to the operating room. They like hearing about it. They don't want to be there. So I think, again, the very people we want to be trained to think about this may not be the people who want to be there. That's why teams are so important. You don't have to be in the operating room. If you like it, you like it. But that it's the idea of tuning a circuit. I see one of the questions is on, well, what about non-invasive stimulation? So I'm all for it if it works. And it's the same way, why take a drug if therapy works? Well, you wouldn't. So it's not a choice. You always want to do the least invasive thing. But if you have a circuit that's broken, you have to go for the treatment that can fix the problem you have. And in TMS, most of our patients have tried TMS, doesn't work. And interestingly, the TMS folks found that the place on the head, on the left side of the frontal, the frontal bone, near the frontal cortex, the best place to stimulate is the place that's most connected to area 25. And they actually use maps to find that location with the logic of you can turn down 25 or you can turn up the frontal cortex. Not if area 25 and the frontal cortex aren't actually working together properly. That may not work. So again, back to Matt's question, you have to know about the person's own brain as well as what's the state of their circuit to actually match up a treatment. But that theoretically, there's new technology being developed that would be deep TMS or other forms of stimulation, even with ultrasound, where you can tune deep in the brain and give it without invading the skull. The question is if you don't fix it, you have to wear something. But if you did it at night, if doing it for a dose at night was what you did, I mean, people take insulin every night. I take a thyroid pill. I don't have a thyroid, so I have to take a thyroid pill every day. I mean, there are ways that a treatment every day could be effective if the technology can be shown to be effective. So I'm quite optimistic, and TMS is something everybody should try first. But in fact, by the time you failed ECT, even I see somebody, I see ECT isn't available in Montana. And for ECT, you have to go to Salt Lake City. You know, I think that if ECT isn't available, the thing after TMS and before like going to Salt Lake for ECT is now ketamine is available. And ketamine in people who work for it's intranasal or intravenous, but intranasal is the clinically available source, and that you know in a couple of doses if it's going to be effective. And when it works very rapidly. And the new use of it is that it used to be you'd get a dose IV intravenously, and if it lasted, it would go away in a week or two. But then when it was developed by Janssen, they were giving it, instead of having to come back, you were taking doses. It worked out a dosing schedule. So I think that for the person asking about, well, what do you do if you can't get ECT? I think that's something that the community should try to ensure that there are providers that are skilled in ketamine. And I'm not saying it doesn't seem like it's that hard. The problem has been getting it into the field, because I know my team in New York who actually did the research on ketamine was, some of the fundamental research on ketamine was done at Sinai. Getting it so that they can deliver it has been slower than one might have thought in terms of getting it going in clinics. But that might be a focus area for the community to make sure that there's access to that.
I have another question for you. I'm a former neuroscientist, but I am wondering, when you're stimulating the white matter, like the cingulate gyrus and so forth, are you being able to understand what the antidromic, what's going back, versus the orthodromic, which is going forward, to what structures, what transmitters, what effects, at the nerve level are taking place? And are you looking at those? Because there can be antidromic effects.
Absolutely.
And
no, absolutely. So here's how we're approaching it.
Really technical for most of our audience.
But I'll try to answer it so that you're satisfied, but other people can follow. So again, I showed the map, the template that we follow with the white matter bundles. One of the things about that kind of map is it doesn't tell you direction. It doesn't tell you area 25 talking to the frontal lobe or to the thalamus or to the cingulate. And when you look at animal maps where they do injections to know the direction of things, everything goes in both directions. So orthodromic, going with traffic along the intended connection of one cell talking to where it's headed, versus backfiring and going backwards down a one-way street, going backwards to something that's coming towards you. And we actually don't know. But there are ways that we can get an idea of how the stimulation is flowing. again, with the caveat that cells in and around area 25 are going to different places and we're hitting them all at the same time. You know, animal modelers can inject cell-specific labels and then stimulate just the specific cells and tease it apart. We can't do that, but there's certainly good evidence that things seem to track together, and how might we look at that? Well, one way you do it, that you could do in an animal that's harder in people, is to have sensors in all the places at the end of all the white matter bundles, so that you'd have multiple sensors in the circuit, and then you could look at the timing. We're not doing that because that would be more invasive, and we can't justify that, but that's certainly something that can be done in animals. What can be done in people is to stimulate at the target and do it slowly and watch how information flows electrically through the circuit with high density EEG or MEG. We don't have magnetic encephalography, but we have a lab, Dr. Waters has a high density EEG system, and she's developed, I didn't put it in the talk, where she can ping right where we stimulate, and she can follow the propagation, the perturbation map of how is information flowing. Can't control the direction, but she can see where it's going. And it looks as though it moves to the front first on one side, then moves to the opposite side, then it sweeps along the back of the head, you know, go from front to back. But when you look at what's the net effect of perturbing the brain, it seems to go kind of backwards and goes into the insula and probably into the mesial temporal cortex. And that's back propagating, because information doesn't flow from area 25 to the hippocampus. That's one of the only connections where it goes from certain parts of the hippocampus to 25. So it may be that learning is slower because the part to the hippocampus isn't really the right way to do it. So maybe if we had a way to avoid that one track, they get better faster. So your question is really profound, because it might be that to get better faster, you need to hit the whole system and then only tune part of it and avoid the rest of it. So it isn't so much about changing the parameters. It may be that you repair, and some things just don't need more stimulation over time. You're kind of fighting with yourself. And I think we'll get there. It's very hard to figure out how to design the experiment to do it. But I think that partners that study animals that have similar anatomy can actually set up ways to do that. I mean, you know, there's teams in Montana that do that in non-human primates, and that's probably the way to do it. Sorry for the long-winded thing, but.
No, and I think that even some of us who are not neuroscientists could follow your description. Thank you.
Okay, good. I mean, you know, it's always hard in a forum like this, even if we're together, because I can see the people on the screen, but not the rest of the audience. Is, you know, you can read an audience and you can watch people falling asleep or on their phone. I can't see everybody if they're like watching another show. I just figured the numbers will go down. But, you know, it's a very hard way to kind of figure out what's the right tone, so and level. So I hope people that are hanging in there are getting their questions answered.
Yeah, well, you know, one question that's came up a couple of times, and it's, you know, being a researcher, it's a question you never want to hear is, well, when is this going to be, you know, a clinical treatment that we can go down and get it at Bozeman?
No problem. So let me talk about that, because again, to have what is a more, I don't know, enjoyable, I don't know is the right question, but more personal discussion, I left out an important thing. I told everybody at the beginning, I consulted to Abbott, and that there's intellectual property to do this, and that Abbott was previously the the technology was previously owned by St. Jude Medical, which is a device company, makes pacemakers. And they ran a clinical trial called the Broaden study that finished, you know, a few years ago. They actually went to take what we'd done in Toronto and move it to a commercial trial before we actually had all, I mean, they drank the Kool-Aid, so to speak, that it looked so straightforward from what we'd done in Toronto. Nothing's ever straightforward. And I think that's what everybody needs to take away. You get one question answered and you got 5 more. But the Broaden study was scheduled to do 200 people. It was a multi-center study around the country. And at 100 patients, the FDA wanted to review how it was going. And when they broke the blind, they found that at 6 months, they had half the people or a third of the people they were implanted, not getting stimulation for the first 6 months. And the other group was getting active stimulation. But they used no tractography. They made an approximation. They had no supportive psychotherapy. It was like all about the machine, and everybody was blinded. And so the percent that got better was 20%. So active didn't beat sham at 6 months, and they decided to stop and to figure out what was going on. On the other hand, those 100 people implanted, then after 6 months had open stimulation and could, you know, go to a therapist. And at a year, it was a 30% response rate. And at 2 years, it was a 50% response rate. So this group of people got better much slower. Remember, I showed that example. Could have been that people were a little off if they'd had tractography. They didn't have that information. So that trial was halted. The patients involved, if they got a good response, they were allowed to get a rechargeable battery and continued like my other study patients have. And if they weren't getting benefit, they were offered to be explanted. And the company is now trying to decide, given that there's all this other research, that they need to try again, because there isn't something for people who fail ECT. Ketamine is a stopgap if it works for you. And I saw somebody did make a note in the Q&A that, and I'll just say it in case other people might have been interested, there's a clinic in Billings who does offer ketamine. So that's good to know for the community. But that we'll hope that Abbott will take everything that they can read. We're certainly consulting to them to encourage them to do it again, because look, people can read our papers and do it off label, but someone has to pay for an expensive procedure. It's not inexpensive. Insurance, we fight with insurance to pay for our experimental subjects. This is the part where fairness and access to get it there. I mean, I can take the attitude, it's not my job. It's my job to do what I can do. No, now it's part of my job to figure out how I encourage the company to do what they need to do the study, to make it available. That doesn't solve the problem of what will be access, but it ought to be in the toolkit of how we take care of people. But this is where doing experiments and doing interesting neuroscience, studies at some point has to be about something real. When you're doing things with people that are sick like this, and we have obligation to take care of people long-term. They have experimental devices. We take care of them beyond the experiment. I mean, that's an ethical obligation. We're going to do this work, then we're responsible. And it's not easy, but as people are well, then insurance, you can twist their arm. Why do I have to twist their arm? Because that's the way the world works. But these are the tough parts. That the science part is easy. The hard part is getting it so people have access. And if you have a suggestion, write Abbott.
Yeah, well, that was a very nice response to that question. And, you know, it really, it was a great example of how all of, you know, from the, you know, as we like to say, from the bench to the bedside, right? There are so many steps between that, you know, just like you say, the science is the easy part. And in so many fields, this is the case. It's all of those other steps that are really the challenges and where things fall off the road, right?
There's a very interesting question that I'd like to answer, asking about kind of doing therapy with stimulation and using positive psychology. So my new team, I've always involved the cognitive behavioral therapists because we really could see that the stimulator did part of it. And as the patient said, you didn't tell me how much work I had to do. I actually will never be convinced that in the broadened study, that if they had done therapy with and without active stim, they would have gotten better. People were just waiting for the device to transport them into something else. I mean, people have been sick 10 years. What are you supposed to do if the door has always been locked and every time you try it, you can't open it, and someone unlocks the door and you never try it to open it, you can never go outside even though you could. It's like that old Twilight Zone episode. The therapy has become essential, and I now have actually a therapist on my research team to actually watch the evolution of that change over time to figure out when is the right time to introduce therapy. And what you notice is once negative is gone, you need to make your own positive. And if you haven't been positive in a long time, you not only over respond to any negative, so you need to repopulate your emotional bandwidth, and you need a guide, you need a therapist, you need to know that having a bad day doesn't mean you're relapsing, but you also need a strategy to test the waters to find things that you can do. You know, if you've been sick even 5 years, in this day and age, you don't go back to your old job. Your old job probably isn't there if you've been out of it for 6 months, much less 5 years, and you're rusty. The positive psychology part, I think, evolves late. The first thing is to get people to just experience emotions that have variance. And I notice that over the course of several months, it's just as though they're getting reacquainted with the range of emotion that are possible. And for a therapist to learn what the dynamics of a person is. Some people are good poker players. You can't tell when they're happy and you can't tell where they're sad. And that's how they naturally are. And other people you can see are probably like me, where I wouldn't even attempt to play poker. I'd be a show all the time. That reactivity has to be relearned. So I think the question is really important. I think they need work camp. If I could bring them all to Montana and they worked on a ranch, that's what they should do for the first 2 months. No thinking, don't think about your bad relationship, do stuff, build up your muscles. Think of it like having a broken leg and having to retrain just weightlifting and some stamina. Then you'll run a marathon. And how we do that with evidence and rigor is, I think, more important now than trying to come up with new parameters.
All right. Well, we're almost out of time here. And so I just wanted to give you an opportunity if there was anything else that you'd like to comment or any other questions that kind of caught your eye.
Okay. So I think, this idea of ketamine and psychedelics, and are they similar? I think they probably are, at least for the first part. What does it mean to get outside yourself, like the patients describe? I think we're watching it happen in seconds. I think when you have a guided trip on psilocybin or even ketamine, people describe they have an out-of-body experience. Sometimes it's pleasant, sometimes it's not. You're out of a rut. The question is when you come off, where do you land? And I think that's an answerable question. And I think time will tell, but experiments need to be done as we have markers that ketamine and psilocybin that will look for things that are similar. I think the field has to get to where they take clues from wherever you get it. Not that every time you start something, you act like it's brand new. Everything is built on something else. And the question is to figure out how they all fit together. So when I read a lot of the questions, you know, there's interest in personalization or, you know, something that's less invasive, or are these things the same? They're similar. And until we understand how they're the same and how they're different by studying them together, I think we will make progress, but we, like every team, it's more of a community team. How do we do it together so that we don't kind of reinvent the wheel? Because there's all kinds of, you know, you don't want to go in circles if you can kind of go straight ahead. And I'm into, you can see I have a big team. Why do I have a big team? Because most of what I need to know, I have no idea how to do. I know how to do PET scans. I know how to watch patients. I don't know how to do the electrophysiology. I don't know how to do a lot of the things that I know are going to give me the answer. So I need to create the environment to find the answer and partner with people that might be studying something else. And I think that's true in the world widely. How do we help each other? Because we all have similar goals. If somebody's got something that they're really dying to know, I think we've covered the general things that I'm seeing. I'm avoiding the engineering questions because I can't answer it. So I apologize. There is, so there's an important question about the motivation of subjects to participate. There's an oxymoron almost of the idea that if you think if you're so ill because you failed everything, do you reach a point where you can't even reach out for help and that you actually can't imagine everything could work? Or do you kind of say, I find that the best patients are those that have a family member help them to reach out for help. If I get an email that's multiple pages long describing like a job interview, that person isn't like Brandy or, you know, the people that you describe where you can't even get your thoughts together. That's a different kind of depression. But I have found, we did a study early on, who in their right mind would volunteer? You know, like, what is that? And we studied the balance between volunteering for a study where you don't know if it will work with actually being so altruistic that you don't mind being a guinea pig. And that being overly altruistic, like, it doesn't matter if I don't get better, I want to help somebody else. I don't believe that, because I think you wouldn't volunteer. That's a different, that's something else. I think it's well-intentioned. People, I'll quote one of the patients, it was patient number 3 in Toronto. You know, people say, maybe people will do anything. And you know, if you said, I cut off your arm, if this other problem was gone, would you do it? And should you be allowed to? No, it's doing no harm. But that the patients read this consent, they know they could have a seizure, they could know they could have a bleed, they could have a heart attack, they could get an infection. You go, boy, that's pretty scary. People go, look, I don't have an option, but what I am afraid of is that I could possibly be worse for having had the surgery, that you hurt me in the surgery, not that I don't get better. I actually never found that patients were worried they wouldn't get better. They were worried that we'd hurt them neurologically, and then they'd be this sick and they'd be disabled in another way. So I remember patient 3, she's on the table, and she had a spot where actually she described that the room brightened up, like the colors became more saturated. It was like the gray filled in. And I said, okay, so are you a little more relaxed now? We're going to do the other side. She goes, no, you just didn't hurt me on this side. I'll wait till we finish the other side and then I'll calm down. So it's very interesting how people approach risk. This noise signal, this distortion of depression, in there is a person saying, help me. And I think if you have a family member who is ill and can't even describe it anymore, that is the right person, and that they need an assist. And that's how motivation is expressed. And I think we just need to listen to people around us who are asking, because sometimes the people that are saying the least are the ones who need our help.
All right.
I'm done talking. You just, you'll have to just hang up, otherwise I'll just kind of
No.
keep going.
Dr. Mayberg, I want to thank you for all that you have said and explained to us. And I say in my native language, getche miigwech. Great, thanks.
Well, thank you.
And thank you, Joe. And let's the rest of us thank Helen for just a really inspiring and thoughtful presentation.
Thanks so much, Helen.
Well, thank you for the invitation. I felt like we were together. I only wish now we were going out for a drink in the bar.
Me too.
We'll hope for another time.
All right.
I enjoyed the opportunity, and I wish everybody in the community well, and we should all be safe, and we'll hope for better times.
Yeah.
You too, Helen. You too. Thank you so much.
Thanks for doing this. This was great.
Yeah.
Okay, well, good night.
Good night.
Yeah.
Bye.
Good night.
Thanks, Brian. Thanks, Bill. Thanks,
Francis.
and
Thanks, Jason.
night.
Thanks, Joe.
Yeah.
Thanks, Joe.